Hello and welcome to this next lesson in our topic on homeostasis principles. In previous lessons, we've looked at the principles of homeostasis, the basics that underpin all the other lessons. We've looked at temperature regulation in both endotherms and ectotherms. In last lesson, we looked at excretion, homeostasis and the liver. And today, we're going to be looking at the structure of the kidney and osmoregulation. So by the end of this lesson, you should be able to define the word osmoregulation and explain why it's important. You should be able to describe the structure of the kidney and the nephron. And you should also be able to explain how parts of the nephron remove waste products and reabsorb useful products from the filtrate. So let's start today's lesson with a keyword. All keywords are always written in green. And the first keyword we're going to get out of the way is the word osmoregulation. Osmoregulation. Okay. Now, in biology, anything that has the word osmo in it usually refers to water. Okay, so think of osmosis. So we can assume that the word osmo means water. Therefore, we can define osmoregulation as the following. It is the homeostatic control of water levels in the blood. So the homeostatic regulation of water levels in the blood. Okay. Now, it's important because it needs to maintain constant osmotic potentials in the blood and tissues. If water levels aren't regulated, the blood can draw water from surrounding tissues and it can cause surrounding cells to shrivel and die. At the same time, we're animals. If water goes from the blood into the surrounding tissues, it can cause cells to expand and burst because they don't have these cell walls to prevent that from happening. They can't become turgid. So we can put underneath here that it's important, and remember, anything in red is a very important point. It is important to maintain water levels in the blood. Sorry, it is important to maintain constant osmotic potential osmotic potentials of the blood and tissues so that cells do not shrivel or burst. So we've said that osmoregulation is the homeostatic regulation of water levels in the blood, and it's important to maintain constant osmotic potentials of the blood and tissues so that cells do not shrivel or burst. Now, the organ in your body that is very important in this job, in maintaining water levels in the blood, is the kidneys. So we can put the subheading, structure of the kidneys. That's not how you spell kidneys. So we'll put kidney. Structure of the kidney, and we can underline it. Now, to sort of break this up a little bit, I'm going to take this picture because it looks good, particularly on the thumbnail for my YouTube videos. And we're going to have a look at how the kidney is sort of structured. So we have two kidneys on each side of our spine. It's found in our lower back, okay? Now, if we were to cut a kidney as a cross-section through the middle, we would have these various different parts. The lighter parts, which are shown here, are known as the cortex, and these are made up of what are known as renal or Bowman capsules, convoluted tubules, and blood vessels. The medulla, which is the darker areas, so the medullas are these sort of triangular dark areas, these are made up of the loops of Henle, collecting ducts, and also blood vessels. We also have the renal pelvis, which would be found down further in here, and the renal pelvis is a funnel-shaped cavity that collects urine. Okay? So, we can get this written down. I'm going to make that picture a bit smaller. If you want to see it, it is on the PowerPoint for you to peruse. We can say that the kidneys, or the kidney, is responsible for maintaining water levels in the blood. So the kidney is responsible for maintaining water levels in the blood. We can say that a section through the kidney shows that it is made of a cross-section through the kidney shows it is made of dot dot. We've got what's known as the fibrous capsule, which is the outer membrane that protects the kidney. So that's not shown on this diagram, but the outer layer of the kidney is known as the fibrous capsule. So we've got the fibrous, F-I-B-R-O-U-S, the fibrous capsule, which is the outer layer of the kidney, that protects it we've got the cortex, which is always the lighter coloured outer region, which I'm going to put in red so that it stands out. So the lighter coloured outer region, if I could spell coloured, the lighter coloured outer region made up of renal, which are also known as Bowman's capsules, renal, R-E-N-A-L, / Bowman's capsules, B-O-W, capital B because it's named after someone, M-A-N-S, after the N it's got a sort of apostrophe at the top because it belongs to someone, capsules. We've got the convoluted tubules and blood vessels. convoluted tubules, and blood vessels. Okay. The darker areas are known as the medulla. The medulla, M-E-D-U-L-L-A, medulla. And these are the darker inner regions of the kidney. The darker inner regions. The darker inner regions made up of loops of Henle, Henle is Henle, collecting ducts, and blood vessels. Okay. The final bits that we need to just describe are the renal pelvis, which is the funnel-shaped cavity that collects urine. So the funnel-shaped cavity that collects urine into the urethra, U-R-E-T-E-R, and finally the ureter is the tube that carries urine to the bladder. Tube that carries urine to the bladder. Okay, so osmoregulation is the homeostatic regulation of water levels in the blood. It's important to maintain constant osmotic potentials of the blood so that tissues don't shrivel or burst. Remember, if you've got too much water in the blood relative to solutes, water will move out to the blood vessels into surrounding tissues and cause those cells to burst. At the same time, if there's too many solutes in the blood, that will cause water to be drawn out of surrounding cells, causing them to shrivel. So it's important that water's kept at this regular level. Water's maintained via the kidney. The kidney is responsible for maintaining water levels in the blood. If we get a kidney and we cut it in half, we do a cross-section. We would have done this in the lab if we were in school, and if I'm honest, it absolutely stinks of urine, so you've probably dodged a bullet there. However, if you do a cross-section through a kidney, it shows that it's made up of the fibrous capsule, which is the outer layer of the kidney that protects it. We've got these cortexes, which are the lighter areas found on the inside of the kidney, and these consist of renal or Bowman's capsules, convoluted tubules, and blood vessels. You've got the medulla, which is these darker sort of triangular areas on this diagram, which are made up of loops of Henle, collecting ducts and blood vessels. You've got the renal pelvis, which is a funnel-shaped cavity that collects urine. And then we've got the ureter, which takes urine to the bladder from the kidney. Now, at A level, you can be given a diagram that is blank like this and asked to label the cortex and the medulla. You can also be given a stained microscope picture or micrograph and asked to label the cortex and the medulla from that. The cortex will always be light in colour, whereas the medulla will always be darker in colour. That is why I've put those in red. Okay. Now, we've spoken about lots of things that perhaps don't make sense to you at the moment, such as the Renal Capsule or Bowman's Capsule, convoluted tubules, loops of Henle, collecting ducts, and we need to talk about what each of these do now. So if you haven't got the notes down yet, please pause the video, because I'm going to refresh the screen and have a swig of my protein shake to keep myself hench. So each kidney consists of about 1 million tubular structures known as nephrons. So I'm going to put in red because it's important. Each kidney consists of about 1 million nephrons. Nephrons. Now, a nephron is a key word. A nephron is defined as the basic functional unit of the kidney. The basic functional unit of the kidney which is responsible for making urine. Okay, so each kidney consists of about 1 million nephrons, and a nephron is the basic functional unit of the kidney which is responsible for making urine. And if we go to our PowerPoint, we've got a diagram of a nephron here. Now, each nephron consists of a renal, or known as a Bowman's capsule, which would be this part here. This is our Bowman's capsule. It would then feed into what's known as a proximal convoluted tubule. We would then have our loop of Henle, followed by our distal convoluted tubule. After the distal convoluted tubule, we've got the collecting duct. Okay. So we need to talk about each part of the nephron and what each part does. If you want that image in a bigger picture, it is on the PowerPoint that I've shared with you. So we can say that each nephron consists of dot dot. We've got the renal or Bowman's capsule, as it may also be known. So we've got the renal or Bowman's capsule, and essentially what this is a closed end of the nephron that surrounds lots of capillaries. Now, these capillaries are known as the glomerulus. So you may also see this called the glomular capsule. Okay? So it may also be known as the glomular capsule. Sorry, I can't say that word very well. I do apologize. So the renal capsule or Bowman's capsule is a closed end of the nephron that surrounds a mass of capillaries known as the glomerulus. Glomerous. Glomerulus. Okay. The inner layer of this Bowman's capsule is made up of specialized cells known as podocytes. So we can say that the inner layer of the capsule is made of podocytes. 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 Okay. After the renal or Bowman's capsule, we've got what's known as the proximal convoluted tubule. So proximal convoluted tubule. Now the word proximal means close to. So it is close to the Bowman's capsule. So this is the convoluted tubule that is closest to the Bowman's capsule. The word convoluted means sort of messy, all over the place. So what it indicates is that it's highly folded. Yeah, it's like a squiggly line. Rather than having a line like that, it's all over the shop like that. Okay. So convoluted means that it's sort of looped around. Now the proximal convoluted tubule, we can say that it is a series of loops surrounded by blood capillaries, a series of loops surrounded by blood capillaries and we can say that the walls are made of epithelial cells with microvilli. So their walls are made of epithelial cells with microvilli. Okay. After we've got this proximal convoluted tubule, so this is our Bowman's capsule, this is the proximal convoluted tubule, you'll see that the nephron drops down and back up. It goes down in this massive U shape. This is your loop of Henle. So our third one is the loop of Henle. L-O-O-P of Henle, H-E-N-L-E. Now the loop of Henle can be defined as a long hairpin loop that extends from the cortex into the medulla and back again. and back again. Now, the loop of Henle is surrounded by capillaries. It's surrounded by capillaries. And it's also worth mentioning here that it has a descending loop that goes from the cortex into the medulla and an ascending part of it that goes from the medulla back into the cortex. So it has a descending arm, which goes from the cortex into the medulla. I'm not going to write it out because of space. And a ascending arm, which goes from the medulla back up into the cortex. So you've got the descending arm of the loop of Henle and the ascending arm. After this, we've got the distal convoluted tubules. The distal convoluted tubules. Now the word distal means far from or further from. So these are the convoluted tubules that are the furthest away from the Bowman's capsule. So distal convoluted tubules are a series of loops surrounded by capillaries. A series of loops surrounded by capillaries. And then finally, down here, we've got the collecting ducts. The collecting duct. And the collecting duct is a tube which multiple distal convoluted tubules feed into. So a tube that multiple distal convoluted tubules feed into. Okay. So each kidney consists of about 1 million nephrons. A nephron is the basic functional unit of the kidney, which is responsible for making urine. We have the renal capsule, which is the closed end of the nephron, which surrounds a lot of capillaries known as the glomerulus. The inner layer of this Bowman's capsule is lined with what are known as podocytes. After the Bowman's capsule, we've got the proximal convoluted tubule. This is a series of loops which are surrounded by blood capillaries, and its walls are made of epithelial cells with microvilli. After the proximal convoluted tubule, we have the loop of Henle. The loop of Henle goes from the cortex down into the medulla and then back up into the cortex. It has a descending arm and an ascending arm. Again, this is surrounded by lots of blood capillaries. The distal convoluted tubule is a series of loops surrounded by capillaries, and the distal one is after the loop of Henle. And the collecting ducts is a duct into which lots of distal convoluted tubules feed into. So if we were thinking about this diagram, we could sort of break this up into where the cortex is and the medulla is. So your cortex would be this upper part, whereas below the line would be the medulla. So anywhere here would be your cortex, and going down will be your medulla. So we get this loop of Henle dipping from the cortex down into the medulla and back up into the cortex. Okay? So we now need to think about how this works. So if you haven't got all the notes, you can pause the video, you can replay it back, and we can put the subheading. Let me just quickly check my PowerPoint. We can put the subheading, the job or the function, because it sounds a bit more professional, because you guys speak way better than I do, the function of the nephron. And we can underline it. Now, just for our reference, I am going to put this diagram over here, and we are going to number it, okay? Just so we've got it. In fact, I'm going to squeeze it up here right out the way so we've got as much space as possible so that we can keep referring back to it. So the first part is what happens here. So number one, we're talking about the Bowman's capsule. So number one, we can say, is the formation of the glomerular filtrate. So formation of the glomular, Glomerular filtrate. Okay. Now, the renal artery brings blood to the kidney, which splits into different arterioles, and then each of which enter a renal capsule. Okay. So you will always have the renal artery go into the afferent arteriole, into the renal capsule or Bowman's capsule, and then into an efferent arteriole. Okay? So we can say that the renal artery brings blood into the kidney, into the afferent arteriole, into the renal/Bowman's capsule, and then blood leaves via the efferent arteriole. So what I'm trying to say here is that we have the renal artery, then we have the afferent arteriole, then we have the renal capsule or Bowman's capsule, and then we have the efferent arteriole. Okay, so that's the route which the blood goes through. Now, the afferent arteriole is wider than the efferent arteriole. So, the afferent arteriole is wider than the efferent. So, this is wider, this is narrower. What this means is that blood pools in this renal capsule because more is entering than is leaving. So the blood remains stuck in the renal capsule. So we can say that the afferent arteriole is wider than the efferent arteriole, so there is a backlog of blood in the renal capsule. Just going to put cap, okay, it means capsule. Now, during this time, while the blood is pooling in the renal capsule, water, glucose, and mineral ions are squeezed out of the blood to form what's known as the glomerular filtrate. Okay? So, we can say, during this time, water, glucose, and mineral ions are squeezed out of the blood to form the glomerular I can't say it, filtrate. I have this problem every year when I teach this, and usually it's refined to my classes. Now I'm recording it. So in this Bowman's capsule, blood moves into it through the renal artery and the afferent arteriole. The efferent arteriole is narrower, so blood gets stuck in the renal capsule. While the blood is stuck in the renal capsule, molecules such as glucose, water, and mineral ions are squeezed out to form what's known as the glomerular filtrate. Okay? Blood cells and proteins are far too big, so they are unable to pass into the glomerular filtrate. So I'm just going to put that blood cells and proteins are too big and do not pass into the glomerular filtrate. Glomerular filtrate. So the blood cells and the proteins can't be squeezed out of the blood. They remain in the blood. Now, podocytes, which line the renal capsule, have gaps between them that allow the glomerular filtrate to pass out. So this is kind of like, the renal capsule is kind of like a sieve. It has these podocytes lining the inside, and between each podocyte there is a gap that allows the glomerular filtrate to pass. So we can say that podocytes lining the renal capsule have gaps between them that allow the glomerular filtrate to pass through. Into the proximal convoluted tubule. Okay. However, the problem is that most of the molecules that pass into the glomerular filtrate are actually useful, and they need to be reabsorbed. Now, that's a really important point, so I'm going to put it in red. Most substances that pass into the glomerular filtrate are really useful and need to be reabsorbed. Right then. So the first stage in the nephron is the formation of the glomerular filtrate. The renal artery brings blood into the kidney through the afferent arteriole into the renal capsule and then leaves via the efferent arteriole. So blood enters through the renal artery, it goes through the afferent arteriole, it goes into the renal capsule and into the efferent arteriole. The important point here is that the afferent arteriole has a wider lumen than the efferent arteriole. Therefore, what this means is the blood remains in the renal capsule. It pools in the renal capsule because it can't leave very quickly. More blood is coming in than it can leave. During this time, water, glucose and mineral ions are squeezed out of the renal capsule to form what's known as the glomerular filtrate. So these molecules leave the blood, okay? Blood cells and proteins are far too big and they remain in the blood. They cannot become part of the glomerular filtrate. Podocytes, which line the inside of the Bowman's capsule, have gaps between them. So if this is my podocyte, and this is another podocyte, and this is another one, there are gaps between these podocytes which allows the glomerular filtrate to move between the gaps. Okay, so the glomerular filtrate can feed through into the proximal convoluted tubule. Most substances that move into the glomerular filtrate and therefore into the proximal convoluted tubule are really useful and therefore need to be reabsorbed. So, let's move on to the next bit. Let the background back to black, as Amy Winehouse said. You guys are probably far too young for that joke. If I move that into the top corner, we've had number 1 taking place at the Bowman's capsule. We're now going to talk about number 2 and what happens in the proximal convoluted tubule. So number 2, reabsorption of glucose. Let's check then. Reabsorption of glucose and water by the proximal convoluted tubule. Okay. Now, 85% of the glomerular filtrate is reabsorbed back into the blood. So that's a really important figure. We can say that 85% of the glomerular filtrate is reabsorbed back into the blood. Okay. Now, epithelial cells that line the proximal convoluted tubule have microvilli that increase their surface area, and they have lots of mitochondria that make ATP for active transport. So, epithelial cells that line the inside of the proximal convoluted tubule epithelial cells that line the inside of the proximal convoluted tubule have microvilli that increase the surface area. In fact, I'm going to stick to that colour for now. That increase the surface area for absorption of glucose and water. So by having a larger surface area, this increases the rate of diffusion and osmosis. Furthermore, the epithelial cells also have lots of mitochondria. So the epithelial cells have lots of mitochondria to make ATP for active transport. Or active transport of glucose and any other molecule that might be useful from the glomerular filtrate. Now, at the same time, these epithelial cells actively transport sodium ions into the blood, meaning that you are making the blood more concentrated. Okay? Therefore, water will want to move out of the glomerular filtrate into the surrounding tissues. Okay? So we can say, and again this is important, so I'm going to put it in red, that epithelial cells actively transport sodium ions, Na+, into the proximal convoluted tubule, to make it more concentrated giving it a lower water potential. Okay, so we're adding sodium ions to make the inside of the proximal convoluted tubule more concentrated, which gives it a lower water potential. Now, these sodium ions are moved into the proximal convoluted tubule via carrier proteins that take in useful substances in exchange, such as glucose. So we can say, oh, don't know why I did that, obviously by accident, we can say that sodium ions, or Na+, flow into epithelial cells through carrier proteins that co-transport useful substances with it. So, for example, you may have, in fact I'm going to use a different colour so it's easier to see, you may have inside of the proximal convoluted tubule, so the PCT, you've got lots of glucose in there that is useful. So these are my glucose molecules. The carrier proteins used would carry glucose molecules, and it would also carry sodium ions. So rather than just transporting sodium ions into the proximal convoluted tubule, so these are my sodium ions, these sodium ions would be transported in and glucose would be transported out as this carrier protein changes shape. So we would move these sodium ions into the proximal convoluted tubule and we would move glucose particles out, for example. Now this would require ATP in order to take place. It would use energy to do that, but it would exchange useful substances for sodium. Now ultimately what is then left is lots of sodium ions inside of the proximal convoluted tubule. So sodium ion concentration is high in the proximal convoluted tubule. Okay, so the second stage in the nephron is the reabsorption of glucose and water by the proximal convoluted tubule. 85% of the glomerular filtrate is reabsorbed back into the blood. Epithelial cells that line the inside of the proximal convoluted tubule have lots of microvilli, which increase their surface area to absorb particles, and they also have lots of mitochondria, which promotes active transport of particles. Now, epithelial cells actively move sodium ions into the proximal convoluted tubule. This makes the concentration inside of the proximal convoluted tubule more concentrated, reducing its water potential. Therefore, water will move out of the proximal convoluted tubule via osmosis. At the same time, as sodium ions are actively transported into the proximal convoluted tubule, useful substances are moved out of the proximal convoluted tubule using carrier proteins. Right then, so that's number 2 done. Let's now think about what happens at the loop of Henle. So we've spoken about what happens at the Bowman's capsule, number 1. We've spoken about what happens at the proximal convoluted tubule, number 2. We're now going to think about what happens in the loop of Henle, number 3. So we can put here, number 3, maintenance. Hope I forgot to spell maintenance, we're not off to a good start. Maintenance of a gradient of sodium ions in the medulla by the loop of Henle. In the medulla by the loop of Henle. Okay. Now, the loop of Henle consists of a descending limb, which has walls that are highly permeable to water, and an ascending limb, which has thick walls that are impermeable to water. So we can say that the descending limb, so you spell descending, you'll have to do, the descending limb is highly permeable to water, and the ascending limb does not allow water to pass, so it is impermeable to water. So the ascending limb is thick and impermeable to water. Okay, so the descending limb is permeable to water, the ascending limb is not. So as we move down the loop of Henle, water leaves the descending limb and more sodium ions enter it. So we can say water leaves the descending limb of the loop of Henle and more sodium ions enter the loop of Henle. Okay, so as you move down the loop of Henle, the amount of water inside of it decreases because it's moving out into the surrounding tissues. However, because we're adding more sodium ions as we go down, the concentration inside increases as you go down, so more water leaves via osmosis. Okay? So for example, if you've got the loop of Henle that's going down, so this is my loop of Henle, and you've got water molecules here, so H2O, that is, let's say, a value of 100 here, but the sodium ion concentration, so Na plus, is 10. Because there's lots of water and very little sodium, water will leave, okay? But by the time you get to the middle, there may now be a concentration of sodium of 50. But water, H2O, may now be at 70, okay? But because there's a greater concentration of this, water will leave. Water will continue to leave out of the loop of Henle. Okay, get rid of all this. Oh, and then we've got the loop of Henle. So, at the ascending limb, so by the time we've got to the ascending limb, sodium ions are actively transported out of the loop of Henle. However, water cannot leave. Water is stuck inside of the ascending limb. So we can say, at the ascending limb, comma, sodium ions, Na plus, are actively transported out of the loop of Henle but water cannot pass. But water cannot cross the impermeable walls. So water is stuck inside the ascending loop of Henle. By the time the collecting ducts reached, sodium ions are no longer found inside of the distal convoluted tubule, which we'll talk about in a moment. The important thing to understand is that a sodium concentration gradient is used to remove most water. So we get this countercurrent between the ascending limb and the descending limb. So through this, if we've got a loop of Henle that looks like this, okay? So this is my amazing loop of Henle. As water moves down the descending limb, it is able to freely leave. Okay? So water is able to leave down its concentration gradient, or from a more dilute area to a more concentrated area. At the same time, sodium ions are pumped into the descending limb. Okay? So in the descending limb, we get these sodium ions being actively transported into the loop of Henle. By the time the water gets over to this end, it can no longer leave the ascending limb. However, sodium ions can. So these sodium ions now leave the ascending limb of the loop of Henle, so that by the time we get to the distal convoluted tubule, water what's left of the water anyway, can leave the nephron. Okay? So we can say, and I'm going to put it in red because it's important, that using a sodium ion concentration gradient twice removes most water from inside the nephron. Okay, so it removes most water from inside the nephron. Countercurrent between the ascending limb and the collecting duct ensures that there is a high concentration gradient. So countercurrent between the ascending limb and the collecting duct ensures that water leaves the nephron. If I'm honest, this is the difficult part of understanding how the nephron works. However, if you're not sure about this, drop me an email or come and see me and I'll go through it with you, okay? For now, you just need to understand that the descending limb is highly permeable to water, the ascending limb is thick and impermeable to water. So as water moves, as the inside glomerular filtrate moves down the descending limb, water is moved out of the loop of Henle, while sodium ions are moved into it. At the ascending limb, water can no longer leave, but sodium ions can. Okay, so whatever little water's left is stuck inside the ascending limb of the loop of Henle. But the sodium ions are removed, so this becomes a dilute solution at that point. Final bit for today, because I have spoken for ages, I am well aware of it, but this is what's on your spec, so there's not a lot I can do about that. Number 4. So we've had the formation of the glomerular filtrate at the renal capsule. Oh, we need to go to orange, don't we? We've had the formation of the glomerular filtrate at the renal capsule. We've had the absorption of water and glucose at the proximal convoluted tubule. We've had further absorption of water in the loop of Henle. We're now going to think about what happens in the distal convoluted tubule and the collecting ducts. So reabsorption of water by the distal convoluted tubule. By the distal convoluted tubule. Okay, so the cells of the distal convoluted tubule have lots of mitochondria. So the cells of the distal convoluted tubule have lots of mitochondria. Okay, This allows active transport of various substances. So we can say allowing lots of active transport of substances Hormones act upon the distal convoluted tubule, which makes it more permeable to water. Okay? So, hormones act on the distal convoluted tubule which makes it more permeable to different substances, and that's all we're going to put. Okay? So the reabsorption of water in the distal convoluted tubule has lots of mitochondria, which allows it to absorb substances, and hormones also act upon this distal convoluted tubule, which makes it more permeable. This is shown by the images that are on the PowerPoint. This essentially explains what happens at the renal capsule. So we've got these podocytes lining the inside of the renal capsule, which allows the glomerular filtrate to be forced out. So the glomerular filtrate moves between these podocytes and goes down into the proximal convoluted tubule. And the other diagram we've got looks like this, and this explains how water is moved by this countercurrent system of sodium ions as it moves from the descending limb of the loop of Henle and the ascending limb of the loop of Henle. Okay, we're starting with a arbitrary value of 300 water. The water moves out of the descending limb. Sodium ions move in. We then have a maximum concentration of sodium ions at number 4, which is represented by the number 1200. As it moves up the ascending limb, we get this removal of sodium ions so that the value is back to 200 when we get back into the cortex. So we've lost 100 arbitrary units of water. Okay? As water moves through the distal convoluted tubule in the collecting ducts, water moves into blood capillaries from an area of from a more dilute concentration inside the collecting tubule into a more concentrated solution outside of it. So, by now you should be able to tell me what the word Osmo Regulation means and why it's important. You should be able to describe the structure of the kidney and the nephron, and you should be able to tell me how the nephron works to filter the blood and reabsorb useful substances. As always, if you have any questions, do not hesitate to get in contact with me via the usual means. I will reply as soon as I can. If not, I will see you in the next video. Thank you very much.